Welcome to this afternoon's session. My name is Hakwan Lau. I just arrived. I'm the chair of this session, which is on scale-free dynamics of the brain and fractal consciousness. So we have our first speaker, who's Biyu, who's from NIH. He's going to give the first talk on scale-free brain activity and consciousness.
I'd like to thank Stuart for organizing this session and for inviting me. It's a tremendous honor to speak here. I do have a disclaimer, which is that I'm not claiming fractal consciousness. It's totally possible, but I don't see that we have much evidence for it so far. So I come from the field of human brain imaging. We look at brain activity in humans recorded by electrodes with neuroimaging. And in my field, we tend to think that brain activity is nothing more than oscillations. So there are all these oscillations at different frequencies, some of which we heard about this morning, that come and go during sleep or anesthesia or during awake state. For example, the first EEG phenomenon ever described, the alpha oscillation, is an occipital-dominated dominant rhythm that comes when you close your eyes. So we tend to think that all these oscillations have different amplitudes and different frequencies. So they sum together to give this sort of random-looking brain squiggles that we record. But if you look at the power of this brain activity at different frequencies by plotting a power spectrum, which basically shows how much power you have at different frequency shown on the x-axis and power on the y-axis. This is in a log-log plot, so we take both the logarithm of power and logarithm of frequency. We get roughly a straight line, and on top of that there are bumps at different frequencies. So disregard this equation if you like. People say anytime you put an equation in a talk, you lose half of the audience. Basically, what this shows is that there appear to be two different phenomena. And these bumps on top of the straight line are the oscillations that the field has been focusing on. And the straight line on the bottom is routinely called the 1/f noise. And the first thing you learn when you come to an EEG lab is to take it out. So what could be this 1/f noise? One possibility is that it is simply engine exhaust. Another possibility is that it's summed from different oscillations. For example, one very dominant theory in the field proposes that the brain does not generate complex noise directly. Instead, it generates a large family of oscillations whose spatial-temporal integration gives rise to the 1/f statistics. So in other words, this field proposes that the brain does not generate 1/f noise. The 1/f noise comes from the investigators averaging over time across many oscillations which come and go. So if you look at short periods, maybe you would say oscillations instead of 1/f noise. If that's the case, we looked at intracranial EEG data recorded from neurosurgical patients. These are patients suffering from epilepsy. So to figure out where the epilepsy is coming from, where the seizures are coming from, the surgeons put in these large grids of electrodes right on the cortical surface to monitor their seizures until they determine where the epileptogenic region is, and then they remove the electrodes and take out the bad brain region. So with this technique, we actually have at the moment, the best recording of human brain activity in terms of the combination of spatial and temporal resolution. So if we look at the power spectrum recorded over long periods of data, so averaged over an hour of data, we see this spectrum that I just described. It's roughly a straight line, which is not very straight, but it follows this slope in the log-log plot. And on top of that, there are oscillations at different frequencies. And the different lines are different electrodes. This red electrode has oscillation at 1 Hertz, the blue electrode has oscillation at 8 Hertz and 20 Hertz, and the black electrode has no discernible oscillation. If we just randomly pick 220-second segment, we find that the 3 electrodes maintain their power spectral shape. And so the scale in this graph is different from the top graph, but the red electrode still has oscillation at 1 Hertz, the blue electrode still has oscillation at 8 and 20 Hertz, and black electrode has no oscillation. And we can also see this different oscillatory pattern or the lack of oscillation in the raw data records. So if the one-over-F noise is not an artifact of the investigators averaging over different oscillations, what could it be? An alternative is that it could be fractals. So what is oscillation and what is fractal? Oscillation, by common sense definition, or if you just open up a dictionary, is something that happens with a regular beat, with a regular speed. And there is a temporal scale to it, whereas fractals is defined by just the opposite, something that does not have a characteristic scale. To give you an intuitive feeling of what this means, if you look at clouds, just by looking at a piece of cloud does not tell you how big it is. You have to compare it to the sun or to the building, which I don't know if you can see from the projector here, to get a sense of roughly how big it is. So in other words, parts look similar like the whole. Many natural objects demonstrate this property, such as fen leaves, snowflakes, mountain ranges, and coastlines. So this property is defined by this one simple equation, and this is, in fact, the mathematical definition of fractal. Let f of x be the shape of an object. Let's say the shape of a piece of cloud, and if we resample that with a factor lambda, the resampled object is identical in its statistical distribution to the original object times a factor. That factor is lambda to the power of h. H is a technical term called Hertz exponent. And this sign, equal sign with a d at the bottom, means equal in statistical distribution. So it doesn't have to be exactly the same, but as long as it looks roughly similar or is statistically similar, such as snowflakes or fen leaves. So now we can take this exact property or definition of fractal and separate out oscillations from fractal dynamics of brain activity. So this is, again, a power spectrum of electrodes from one epilepsy patient recorded intracranially, meaning the electrode is right on the cortical surface. And these are from the awake state, and these traces are from the slow wave sleep. And so if the signal is fractal, then if I resample it, the power spectrum would look similar. But if the signal is oscillation, when I resample it, it would become a different frequency. So by computing the cross spectrum, I would have nothing. By using this method, we can separate out the fractal component from the oscillatory component of the EEG activity. So EEG signal and many electrical or magnetic signals recorded from the brain is composed of scale-free dynamics and oscillations. The two can not only be separated, but they are in fact as different as they can be. One is defined by having a temporal scale, and the other is defined by the lack of any particular scale. So when I first presented these results to my colleagues in cognitive neuroscience, the most common objection I encountered was, if the 1/F noise is present in everything from Earth to Sun, how could it possibly be important for the brain? So the question often comes in the form such as, surely the Earth or the Sun is not conscious, but the brain is. Then if the 1/F noise is important for the brain, it must be different from the brain to the Earth or to the Sun. So we looked into the signal and tried to see whether there are structures in this signal. Even if the power spectra look similar, maybe the fine spatial temporal structures of the signal could be different. So indeed, we found that there is extensive phase amplitude coupling. What this means is that for different frequency bands in this scale-free activity, I extract the phase of the low frequency and the amplitude of high frequency. And the color plotted here shows the percentage of electrodes with significant cross-frequency coupling. So this might not mean much to you, just mathematical terms, but in fact, we can use this tool to uncover different structures in different signals. We went on to do similar analysis in Earth's seismic waves and stock market, both of which have a power spectrum that look very similar to the one from the brain. The slope of the brain activity, Earth activity, and stock market all have a slope of -2 in the log-log plot. You can almost superimpose them on each other. So the Earth wave and stock market also have nested frequency patterns, but the exact nested pattern is different from the brain to the Earth to the sun. So in Earth activity, I don't know if you can see the blue trace down here, it's often a bimodal distribution, and in the stock market it's more random, whereas in the brain it's always a unimodal distribution. We have some inkling about what these different patterns mean, and we are still following up on this. But this basically says that even if the power spectrum looks similar, the so-called 1/f noise can have very complex structures in different systems, and these structures can be different from one system to another. And just to mention, some physicists might think the 1/f squared noise, if the slope is -2, is just a Brownian motion, and Brownian motion is purely random. So in fact, when you do this nested frequency analysis on Brownian motion, there is no structure whatsoever. So the next question is, would this 1/f activity, or scale-free brain activity, be involved when you do a behavioral task? So we showed that this scale-free brain activity can be altered when you perform a task only in the brain regions that are involved in the task. So we asked patients to do a simple visual motor task. When something on the screen changed, they press the button and try to do that as quickly as possible. We found that when they performed the task, the slope of the scale-free activity became shallower. So in technical terms, that means that the power law exponent decreases. What that means is that the signal contains less temporal memory or temporal redundancy, because the higher the slope, there is more correlation in the signal with its past. Whereas the decrease of the slope means that the signal is more efficient in encoding online information. So this is consistent with our idea that the brain needs to remember the past and imagine the future in the spontaneous state, and it needs to process information efficiently in the task state. We went on to do similar analysis with functional neural imaging data, because with functional neural imaging, we can look at the whole brain, including the deep structures. And we found that it also shows a 1/f slope in the log-log plot. It's a straight line, and the exponent is around -0.8, a bit different from electrical activity, which is potentially due to the neurovascular coupling, because the fMRI signal measures a blood flow related signal. So this is just different models showing that the power law model is the best model describing the signal. So we even went on to show that this exponent, which indexes the amount of redundancy in the signal, is different in different brain networks. So with how steep the exponent is, we can pull out different brain networks that Melanie talked about this morning in the resting state study. And the brain regions that are more expensive metabolically have a higher power law exponent, meaning that it's more correlated over time. So these are different dots at different brain regions, and on the x-axis is the glucose metabolism of the region. So the higher the metabolism, the larger the power law exponent. Further, we can use similar method to differentiate between people with high or low trait anxiety, or between Alzheimer's patients and age-matched controls. I won't go into the details of how these metrics were computed. So I hope this leaves you with the notion that the 1/f noise is not really noise, but it's a meaningful activity to the brain. So let's go back to the power spectrum. The higher frequency range of the power spectrum is correlated with neuronal spiking rate. So as neurons fire more and more, the activity becomes, in this broadband activity, goes up. So in the remaining session, I want to focus on the low frequency of this 1/f spectrum, which is called the slow cortical potentials. It's I'm going to walk you through the following characteristics. It's an index of layer 2-3 neuronal activity. It correlates with fMRI signal, which I actually won't mention. And the phase of slow potential modulates higher frequency power and neuronal spiking activity, behavior performance. So a series of elegant physiological studies in the 80s and 90s had shown that the superficial layers of the cortex, when you're excited, it causes a long-lasting negative potential that can be recorded with EEG, our intracranial electrodes that we used. And this is the basis of this slow cortical potential, the low-frequency component of the power spectrum I just mentioned. And the superficial layers of the cortex is where the feedback connections preferentially terminate, which recalls George and Melanie's talk this morning. But it's also where the non-specific thalamic nucleus, the non-specific thalamic pathways terminate, and where there are a lot of long-range horizontal cortical connections. Another interesting thing, which also resonates with George's talk this morning, is that acetylcholine modulates slow potential very strongly. And George mentioned this morning that acetylcholine appears to be correlated with a state of conscious awareness. Unfortunately, because it's technically hard to record the slow cortical potential, it's been neglected in a lot of modern experiments. But as Dr. Goldring, a neurosurgeon at WashU, mentioned in the 70s, if DC EEG recording is used, virtually every stimulus-bound cortical activity is seen to be accompanied by a change in cortical steady potentials. And so now the real-time experiments is in fashion again. In fact, there was a series of elegant studies showing that if you present stimulus at negative or positive shift of the slow potential, humans or monkeys can perform tasks differently. So if you present it on the negative shift, you get faster reaction time and better accuracy, and vice versa. And at the negative shift of the slow potential, you also have lower threshold for conscious visual perception. Based on a range of experiments, we proposed a hypothesis suggesting that long-lasting synaptic activities in superficial layers manifesting as slow potentials in surface recording are low-frequency current source density activity, which is just a type of recording brain activity in superficial layers, carry large-scale information integration in the brain and contribute directly to conscious awareness. I'd like to stress here that I'm not suggesting that slow potential per se is the basis for conscious awareness, but only that it indexes neuronal activity in superficial layers, which we think directly contribute to conscious awareness. So as like many theories in the field, there are a lot of details to be worked out, but it appears to be broadly consistent with existing experimental evidence. For example, in threshold visual perception, if you give a very weak visual stimulus, when the stimulus is perceived, there is a long-lasting slow negative potential, shown in black here, between the stimulus onset and motor response. And when the stimulus is not perceived, this potential is gone. And similar phenomenon has been described with intracranial EEG by Stan DeHaan's group. And many, also as George and Melanie mentioned this morning, many EEG ERP phenomenon that correlates with conscious perception appear to be late latency. So normally it's a few hundred milliseconds as opposed to within 100 millisecond range. This is just one example, which is N400 involved in semantic analysis. Libet's work has been controversial in this field, but I think he did one of the most elegant series of experimental studies. He showed that with cortical stimulation, if you use threshold stimulation, it has to be above around 500 milliseconds for it to elicit conscious perception. Whereas if you go below that duration, you have to increase the intensity of the stimulation dramatically. So what this means is that this increase of stimulation intensity will probably cause reverberation of cortical excitation within the cortex, which will last longer than the externally applied current. And this experiment has been replicated by recent study and extended from the somatosensory domain to the visual domain. And as many of you know, the branch-out potential is, which precedes your self-initiated action, is part of the slow potential. George mentioned this morning that in anesthesia, in the loss of consciousness, the long latency ERP disappears. This separate study showed that this long latency activity that disappears during anesthesia persists at 1 second or, in fact, 2 second range. So before the induction of anesthesia, you get first the triphasic ERP to the auditory stimulation, followed by a long-lasting negative slow potential that persists for more than 2 seconds. And during the loss of consciousness and anesthesia, the primary evoked response is maintained, whereas the negative potential is gone at a longer latency. So I mentioned earlier that the slow potential is generated mainly by superficial layer neuronal activity, and also that the non-specific thalamic pathways terminate preferentially in superficial layers, especially layer 1. And recently, the beautiful study from Nick Schiff's group showed that stimulating of the non-specific thalamic nucleus can recover behavioral response in vegetative patient. And the stimulation of this pathway would increase excitability in the superficial layers and cause negative slow potential in those regions. In those regions. And just to mention that similar phenomenon has been described by a German neurosurgeon in the 1970s. From an evolutionary perspective, the superficial layers of the cortex is where the thickness expanded most throughout evolution. So these are the layer thickness of layer 1 to 3 down to layer 6 in the cortex of human, rat, and mouse. So in humans, the superficial layer really expanded the most. And if we look at the EEG activity, and I'm sure Peter will talk about this more, of invertebrates versus vertebrate, the invertebrate activity, brain activity, is dominated by fast activity. So the EEG looks much more spiky, whereas in vertebrate, there is a lot more low-frequency activity. In fact, I didn't put in this slide. If you look at the power spectrum, the vertebrate shows a 1/f type power spectrum, whereas the invertebrate looks a lot more like white noise. So it has been suggested that maybe the slow potential is too slow for conscious awareness. I just like to also mention that, in fact, from decades of psychophysics study, we know that consciousness itself is rather slow, and the amount of information that can be processed by conscious awareness at any one instant has a pretty low bandwidth. Recently, a study led by Spoolmaker et al. Showed that with resting state fMRI activity, you can delineate these brain networks which are much reduced when you fall into deep sleep. So in light sleep, all of these regions are much more connected as compared to deep sleep. And this also recalls the talks this morning, but I'd just like to mention that because this study was done with resting state fMRI signal, and because the fMRI signal itself is even slower than slow potential, this means that with this kind of activity, we can in fact differentiate between different states of conscious awareness. And the authors concluded that our results may be relevant to the debate over whether low-frequency both signal allow us to describe changes in conscious awareness and underline the important role of intrinsic human brain organization in such dynamic phenomena as sleep. So I'd just like to briefly mention the relation between slow potential and other postulated neurocorrelate of conscious awareness. For example, the P300 which has been suggested as a neural correlate of conscious awareness, may be the flip side of the negative slow potential. So the P300, especially the late component, which happens between 300 and 600 milliseconds, appears to increase when the stimulus is increasingly entering the conscious awareness. In this particular picture, the x-axis shows the latency between stimulus and the visual mask. It's a visual masking task, and as this latency becomes longer, more and more stimulus, the stimulus gets into the conscious awareness at a larger fraction. And along with that, the P3 amplitude increases. So if we go back to the model, when the brain let's say this is a hypocolumn, when it is excited, if it inhabits surrounding regions, which is often needed in the update of working memory, that will cause positive going potential also at the long latency in the neighboring cortical regions. So this may be a mechanism for the P300 evoked potential. And the slow potential phase modulates higher frequency activity, such as gamma band activity. Some people might prefer to call gamma oscillations and neuronal spiking. So in this unpublished observation, we found that when the slow potential is going towards a negative phase, meaning that there is more cortical excitation, there is increased amplitude of higher frequency activity from 50 all the way to 150 hertz. And earlier study has shown that at a negative phase of the slow potential, there is increased multi-unit activity. So in the remaining 2 minutes, I'd like to just mention some preliminary data we have done trying to study visual threshold with MEG. So this work was done by Qi Li, a very talented postdoc in the lab. So we used a paradigm in which we probed for both subjective perception and objective performance. So in every single trial, first the subject fixates, and then after a random blank, the stimulus comes down, which is a Gabor patch. I don't know if you can see it from back there. The Gabor patch is tilted to left or to the right with equal probability, and its duration is titrated to be the subject's subjective threshold, meaning that at 50% of the trials, the subject sees the pattern, and 50% of the trials they don't. And then after random delay, we ask, was a patch tilted to the left or right is forced alternative choice task, and did you see the pattern or not? So in this way, because we have both subjective measure and objective measure in every single trial, we can tease their effect apart in the brain activity. So consistent with earlier work, we found that the forced alternative choice performance is better than the subjective perception. And also, when they see the pattern, their performance is better. So we looked at the MEG signal at averaged over 10 millisecond window from stimulus onset, shown by the red line here, all the way to 3 seconds after the stimulus onset. And we found that the MEG activity differentiated between the trials where the subject saw the stimulus and those where they didn't, all the way up to 3 seconds after the stimulus onset. Whereas for the objective performance, meaning that whether they got it right or wrong about the stimulus orientation, there is no such long-lasting phenomenon. So because I think a lot of earlier work may have the confound of when you see something, the performance is better. So by dissociating between these two phenomena, I think we got pretty good evidence that the slow potential correlates with subjective perception, but not with objective performance. So let me just thank my collaborators at Washington University, Mark Rico, Avi Schneider, John Zampo, and Matt Smith, and the patients and clinical staff at the St. Louis Children's Hospital and Barnes Street Hospital, as well as the members of my lab now, Chile, the person that did the MEG study. And thanks Stuart again for organizing the session. I'd like to take your questions.
Frequency is recorded.
Maximum
Sorry.
frequency in a human being that has been recorded by you.
Okay, so if we are talking about the field potential with local field potential, depth electrode, or the electrode I just mentioned, the reliable recording is up to 200 hertz. So if you go above that, it becomes tricky whether you would get contamination from potentially the artifacts or just whether they. So I think it's a limit that hasn't been well tested. Some people.
I think that there should be a limit of the human radiations of gamma, which should be actually to be scaled, because the super consciousness has to be coming in between. In somebody is enlightened or experienced, what should be the gamma radiation coming out from his body? This is my question.
So about gamma, there is two separate gammas. One is the gamma oscillation, which appears to occur around 40 to 60 hertz, and doesn't go much above that. And then there is a broadband gamma in this scale-free spectrum, which correlates strongly with spiking. So if you record local field potential, in fact, when people do those monkey physiology studies, they filter the same signal in the high frequency range to get the spiking activity and the low frequency range to get the local field potential. So if you go to that very high range, you are going to be contaminated by spiking rate.
Okay.
And it's very hard to tell them apart.
Thank you.
Sure.
Do we have a second question? Yes. Didn't realize I had to run to Mike.
Thank you for a wonderful talk. I had a question in terms of the association of scale-free phenomena and consciousness. You brought up slow-wave sleep. REM sleep, however, as far as I understand, doesn't have a scale-free or power law behavior. It's Gaussian. And there are also phenomena under anesthesia or unconscious states that do show scale-free patterns. So I was just wondering if you could comment on that.
Yeah, actually, in the power spectrum I showed, I had awake and slow-wave sleep. Unfortunately, I didn't have the REM sleep, but we published that in the paper. And in the REM sleep, the power spectrum look identical to awake in the scale-free component. The specific oscillations might be different, but it look very similar to awake state.
It's just the theta, actually, that seems to have a Gaussian distribution. But I think there are past data suggesting that those state transitions were actually Gaussian and didn't have good state memory, which would suggest power law. But I'll have to
So
review
maybe
your
you're
work
talking
again.
about the distribution of a particular frequency band, like whether theta itself is scale-free or Gaussian. Okay, so I guess over here it's just a raw voltage trace, and you do the power spectrum on that. Yeah.
Okay, let's give the speaker one round of applause once more.